So what about the calculations? Group wise is ready. Where's the group number 1? You have the diary, right?
So, liquid chemical.
Okay.
We are actually like I took 0.1.
So from where you will get this chemical?
From the chemical.
Have you asked?
Yes.
To whom? Okay, he has allowed for it.
So that today we get from
Okay. About?
this is also this.
See, he will give. He was telling about the other metals, that metal salts. Maybe that is like we may go for zinc cobalt and so many others where he was telling. So ask him if it is possible. We may go for the mixture. Zinc sulfate. Here is zinc sulfate. Okay, that is like a so chlorides. All the that is like salts. Okay, that is like when we are looking sodium chloride. Okay, so metal chlorides all are there. So you can go for the one group of elements in their chlorides itself. Okay, they are the salt form. So we may go for the chemicals. So he was just telling that whenever you people are coming, then come to me. We may go for it. Okay, so in total, what about the other group? Group number?
Like for me, I 50 ml water. 50 I think that would be.
Yeah, it will be later. It is not a first part. It is on a second step. How many?
So much.
Yeah.
We did not make too much.
Ah, yes.
So it's from 50 ml.
So you are looking for preparing 0.1 normal. Okay, here it is. How much?
Here also I took the same amount. 50 ml.
Okay, so how you calculate 0.1 normal?
First, we need to go
Small weight. Okay.
Like when this was in ionic form, it produces 3 ions, electrons. So the thing, so it is coming up to the equivalent distance 52.7.
Equivalent weight in
To prepare 50
let me see that. Yes, group number 1, Mitsu, Nancy, Nisha, Kushi. Who is there? Anybody? Okay. Very good. So anybody has just calculated how many that a liquid broth will be required and the solid? Just I have written as a liquid. One may go for the directly the whole total preparations. It is not like a 10 ml. It's like a 10 ml. It is there. Okay. So I just told that if we go about 4 chemicals, so 4 tubes will be required. So 40 ml. If you are having the different varieties here, so maybe it's a 200 nutrient broth is required to prepare, nothing else. During this, each tube we need to do other preparations part. Okay. So I have written like a liquid and solid both. Okay. So that is why I thought that first to discuss, then I may take you in a lab. Otherwise, it's the wastage of time for me and you people. Always it is. Otherwise. So whether we are going for the temperature, pH, whatever it is there. Okay, I just told about the temperature. Okay, so temperature, maybe it's a first group. Okay, what are the different parameters we are looking into it? Maybe a certain at a 0 degree, 4 to 8 degree centigrade, 25, and 37. Maximum up to this we are going. So 4 tubes will be prepared. Number of the tubes will be 4. So okay, so number of the tubes is going to be 4. First group, first part. Then the second, it is related to pH. Second number of group number 1, and their activity. Okay, so what we are looking to do here, we may go for a range. So you need to prepare 0.1 normal HCl, 0.1 normal NaOH. Okay, by using it, you may be knowing that how to set the pH. Have you done any a time? Yes or no? Not done? Yet not done? Never. Okay, fine. I think we are having the pH rods and meters. So what we can do? Do you know how to calculate 0.1 normal HCl first? Can we read that? How to prepare 1 normal HCl and 1 normal NaOH? Do you know? We are looking for normal or molar. That is the equivalent weight in a 1000 ml. Do you remember such things? Here it is a 1 HCl, so plus and minus. So it is, we are looking for what? Na plus OH minus. Right. So molecular weight, it's going to be remain same for the normal and the molar concentration. It's going to remain same for both. So you need to prepare first what? These solutions. Okay, 0.1 normal NaOH, 0.1 normal HCl. Solutions you need to prepare. Maybe you are going to prepare only 100 ml. So first of all, you are going to prepare 100 ml of 0.1 normal HCl, 0.1 normal NaOH. By using it on the pH meter or the pH rod we are going to have, I have to check in the labs availability. Okay, so you need to prepare a solution. So you may prepare in a 20 ml, about a 20 ml on a nutrient broth. So whenever you are preparing any sort of the solutions, maybe in a beaker, while preparing the pH or the setting the pH, we need to take care of first that we are going to make up the volume 20 ml. So you need to take about a 15 ml of the solution and then make up till a 20. Generally, when you're looking for, by adding the 0.9 HCl, NaOH, maybe 1 or 2 drops, it's going to reduce the pH very drastically. Okay, so it is like when you are performing it, each beaker of flask you are going to prepare, it will be very less amount will be required to add. So you may use the different smaller pipettes are there, 100 microliter. You made the 100 microliter, add into it, and you are going to measure the pH. But right now, it may be not that much feasible because we are not looking to do every sort of the experiment, and it will become more detailed. So what I will suggest is, please, that rather to go for a whole, otherwise I was planning to do in different format that pH range. Okay, pH may be 2, 4. Okay, otherwise we may go for the 2, 4 or continuous 2, 3, 4, 5, 6, 7, 8, 9, and 10. I was looking to perform in a way that this way. So when you are doing your inner tubes, okay, on the next day when you are coming, we may go for the turbidometric analysis. The spectrophotometry we may perform, and you are going to get the ODs. I was otherwise planning for it. So what we can do, that is a pH 2, 4 to till it is there. What was the OD? Accordingly, we are going to gather the growth. So generally at a 7, it will be higher. So it will bell-shaped curve can be possible. Okay, but if you are looking for doing such experimental part, you need to handle the pH meter properly. If anybody has done it, pH meter related part? Yet not. So it will be more difficult for me because if you are looking to do so many experiments together, we cannot go for it. Okay, but in we are having not that much time. Second problem, right now it's about 30th of the September, right? By before 30th October, we are going to have our exam completed, syllabus completions, everything in journals. About the 15th of October, near to all timetable will be start to given to you. Between the 15th to 30th October, let me be chemistry, biochemistry and physiological practical exams is going to be there. Maybe for it may be scheduled for 1 or 2 days. So before that, before 15th October, you must have your journal completion for each subject about it. Sometimes the faculty may allow for the 20, 25. On the next day, on next day it is maybe your practical viva, and before that you may going to complete your journals. It depends upon others. So what I am targeting, that is our 15th of October, our journal should be get completed. It must complete. Okay. So we have just started to write it down. Okay. So it will be a more time consuming. That is why I told that today we are going to perform as simple as possible experimental part. And then after, if the time will be permitted, we may go. Because maybe it's the 14th of November. Between somewhere, your final exams will be there. So then after, you are not going to have much time. You will be busy in your theories. In between, in October month, you are busy with your journals and practical completions. Right. So time is not much permitting me for doing such so. Right. Because we have gone for so many repetitions. That easier way we have completed. So these are an additional aspects. If that only 1 or 2 experiments were there, I have gone up to 7 to 6 to 8. Okay, so we may not going for a whole range. Yes, in exams I may give in different format, or that a table will be given to you. Prepare the chart or the graph of, like here it may be given. It is like a pH. Okay, 2 to 10 is there, and optical density. Prepare the chart or the graph of the given pH to OD. I think that you can do. Okay. Yes, what direct reading may be given to you. It is like a 25 marks or 50 marks pattern is there. We are going to perform 1 or 2 experimental part, and theoretical questions can be asked including this. Okay, direct reading may be given to you, and then you have to follow. So it is the way same we are going to perform in a way. So right now, I think it will be not much permitted to us. We may differently go for this. Otherwise, our whole experimental part, what we are today looking and understanding, won't complete. So pH balance and maintenance, we may go for in a next semester. Yeah, we are going to perform on the growth curve. After this, on the next study, we will perform the growth curve. That day, you are going to perform spectrophotometry compulsorily. Okay. So right now, I'm not looking for it. We may go for the pH directly. That is a NOH flex. You may just add one flex. One, when you are opening it, one flex you have to. It is like a one small cubes, sugar cubes like this. It will be round cubes are there. That is called the flex. Just add one NOH flex. So it is going to be a alkaline. We are not having much time, so I'm not looking to take you in that part. Okay, one is you may go for the NOH flex. Another it's also HCl. We may go for the some diluted HCl. Diluted HCl, and we may add maybe it's a 20 microliter about it into one tube. So one became very acidic, one is became very basic conditions, and that our E. coli is able to grow or not. Okay, the subdivided experimental part we may go. So here it is how much tube? In temperature range, that is the 4 tubes will be there. Here we are going to have at least 2 tubes. For you? 0, freeze. Freezer ma muki diyo. 4 to 8 in refrigerator. Okay, 25. Now that is you may go for here. It may be included as a room temperature if you wish. Then the 4+1. Incubator માં છે. This one is also incubator. This one is also an incubator. नहीं આ અલગ છે ને 25 વાળું ઇન્ક્યુબેટર છે આપણી પાસે 25 વાળું છે 37 વાળું છે. So it can be go for the 5 tubes. Okay. Then the third group, what I have this thought. Yeah, different chemicals. Okay. So in it also, we may go for the preparing the solution. Okay. Or else you may add pinch of it. Pinch of chemicals. I think he has asked about 1 or 2 chemicals to our there in a Gentisar or someone. So I may go for the 4 to 5 tubes. It is required to prepare in a format, but here we are not looking to do. So it's like a pinch of chemical you may have add. Either it's a pinch or a certain about animals. We may go for 40 to 50 microliter if it is a liquid. Here we may go for either like a camenophore, ZNS, zinc sulfide, different chemicals. We are just looking that is it going to disturb or not. Something is there if you are going to add into the tubes. So we need to add before we complete autoclaving. Before autoclaving we have to add. So as much as chemical you are able to get it, go in chemistry lab, prepare, get the 4 or 5 tubes, and broth is prepared 20 ml. Sorry, we are looking for the 10 ml only. In a tube, 10 ml you may prepare, just add the pinch of it. પછી આપણે શું કરશું? We are going to do autoclaving. Then we after incubate, before incubations we are inoculating. Yes, that's the major important part. So on a next day, after the incubations, what we are able to observe with a normal. Okay, which normal is there any growth variation is there or not? So on a Friday we will observe it. So today various different aspects are there. We are going to inoculate also. Okay, so it may take a 2 or 3 o'clock, maybe minimum. It will be time requirement. That's why I told that come before with some calculations. But I was hoping that my after the lecture or one lecture will be divided about it. Then only about at 10:30 we may able to start. Right. Number 4. So here different chemicals. If you are going in lab, I have just talked with the Jayanti sir that you also may go. As you people are using some different organic or inorganic chemicals or salts, just some selected. What you are adding, just write it down in your diaries. Fine. Number 4. Yeah, different metals or solids. So here, for that we may going for the Enaga pledge. In Enaga plates, what we are going to do, we may go for the some sector 1, 2, 3, 4 as much as possible. Okay, and you may use copper or the aluminum foil is possible. Okay, copper plates may be there, smaller. Sheets are coming. Maybe you are using in your laboratory, right? Just we need 2 or 3, not more than that. So how we are going to put over it? Here it be based on the plate. So this copper, aluminum foil or any powder, metal ionic powder is if it is given, we are going to take pinch wise into one plates. Differently, we are going to it's required to sterilize it. Sterile, this one is required. Okay. So differently in blank plates, in Petri plates, or in either in a tubes, either in a plate or in a tube, you are going to make it sterile. Okay, what then after you are going to do? Heating is going to disrupt. It may melt. Chemical cat melts, and then the effect may be not as what we thought may come. So on Enaga plate, you are going to do a spreading first. After the spreading, so you require to put the spreader also to be get autoclaved, right? Clean. Either you may autoclave, otherwise you may just on the alcohol dipping, you may follow. Okay. So Enaga plates, spreading is done. make a 4 sector, and maybe it's a different size copper plates are available to you. Just put over it, okay, and just push it using your spreader. So what's going to happen on the next day, if the copper is may effect on our bacteria, so lawn growth will be not there, it may get inhibited. So it is our aim. So effect of the metals on our bacterial growth. So it is a, yes. આ બધા કેસમાં શું થાય છે, we are going to add from the beginning. pH માં તો કઈ લેવા દેવા ટેમ્પરેચર માં નથી, pH ની અંદર થશે. We are going to add initially this chemical into it. In the second, in chemicals, pinch of chemicals, we are going to add from the beginning, so it gets dry. But here it's not. Okay, fifth variation. Seven to eight. Number 4, number 5. Yeah, antibiotic. So I will provide the strips. Strips will be there. It is like a either square or the block, rectangle blocks will be there, or I may provide the some round strip. Okay, but unlike strips may be possible. We do have in our laboratory. Just I need to check. So what we need to do? We need to go for the agar plate. Simple. Okay, so you may prepare at least here how many? As the metal solid will be there, you may go for the 3 to 4 plates together. Here also you may prepare 3 or 4 plates, not more than that. Yeah, number 6, it is salt concentration. Here we are going to go for the NaCl. Okay, so here it will be go for its range in a percent. How to calculate the percent? Please pardon, I'm not able to get. Yes, sir. Oh, still it is there? I think it's over. About to get over.
It is the starting time.
Oh my God. So give me 1 or 2 minutes. I was not knowing. Okay. Just 2 minutes. Sir. Yes. Sure. I thought that is completed.
No.
Still it is there? Tomorrow?
Yeah, 9 o'clock to 10.
No. Tomorrow?
Tomorrow.
Because the message came to me that is till the Wednesday. So I came directly.
No.
It's okay, sir. Yeah. Okay. So here we may go for 0 to 5% salt concentration. Just take one snap. We may go directly in our lab. We'll discuss more further there. Let's vacant our. હજી એક બે છે. આ લોકોને ઓલું લેવું છે લેબ. ત્યાં જઈને કરશું. હાલો. Let's move in a lab.